So over here we have a series of peristaltic pumps, and I use these pumps to filter water onto little tiny filters. So I put the water bottles over here, up to 4 liters, and I pump them through here, and it comes out here, which are usually attached to the filters. The filters are then size fractionated in 3 and 0.2 microns so that we can get a different idea of which sizes have which different species in them, for example. And then from there they get flash frozen in liquid nitrogen, and then they'll come back to my lab with me, and we will do a DNA extraction and amplification and send them for sequencing. Over here, this is where like the filters come to get organized before they get put into little vials before they're frozen, and I also am working on a DNA extraction here because we have a little, we call it InNavi protocol to see if there's NIFH. So that gets all set up here, and then it gets put over here, which is this is our onboard qPCR, which is quantitative PCR. So we'll get kind of a basic idea if there's NIFH or nitrogenase present in the water. Yeah, and then that's pretty much it for me, besides taking the water off the CTD. Oh, there's also the fume hood. Want to see the fume hood? So this is the fume hood, and here I'm working with formaldehyde, and that's why we have to be inside the fume hood. And what I do here is take probably 95 milliliters of the samples that I'm filtering in there and the samples from the Sippler group, and I fix them with the formaldehyde, and then I filter them through here so they'll be on small polycarbonate filters. And then with this you can look at them with a microscope, such as cardfish or genefish. Yeah, and so that way we'll be able to identify and actually like see who we're looking at. Where on the other side we'll just get sequences, so you never actually get to see like what they look like. Yeah, and I think that's it.